Indeed a great pleasure to be introducing Able Lawrence, a great and dear friend. For the past many years, I've known Able in different capacities, and starting off as a student when I was training in Sanjay Gandhi Institute. And over the years, our families have grown very close to each other. And I must say that Nisha is absolutely right when she said, as a walking encyclopedia, you bring any topic under the sun and he would have volumes to discuss on it. So if you're going to ask him any question, make sure your calendar is free for the next little while, because he likes to talk a lot and he knows a lot. So Abel is from the 33rd batch of Calicut Medical College. After his MBBS, he went on to do his MD medicine from PGI Chandigarh, and after that did his DM from the Sanjay Gandhi Institute in Lucknow, where he currently serves as professor of immunology and rheumatology. He is quite famous in India, and the go-to person for a lot of journalists who want to know the true state of affairs, whether it is regarding the virus, where the epidemic is heading, how is the testing happening, or what is the status of the vaccines. He is the go-to person for most of the journalists there, and he's a frequent face on multiple televisions. He's a big force on Twitter with many famous followers, including our President Obama himself is a follower of Able Lawrence. He predicted this pandemic long before any of us were talking about this virus, and he's quite famous for that on Twitter as well. So without further ado, pleasure, Dr. Lawrence, to have you here. Able, take it away. Well, that is something which I missed. He is an avid wildlife photographer and a bird photographer. Look at that.
I would say that Nejil is my guru
he
for
has
the
documented
bird.
the maximum number of birds in this SGPGIMS campus, which is now considered a hotspot in Lucknow. Able, are you mute?
No, I don't think so. Can you hear me? Hello,
it's
can
very
you hear
early
me?
in the morning. I know it's I think
I'm not
6 o'clock there in the morning.
We can hear you.
Yeah, we have now are in the golden age of vaccines. The one good thing that coronavirus has done is that all the vaccine technologies hiding under the drawers are coming out.
Can't hear you. I don't think your audio is activated. Did you activate audio when you logged in?
Yes, it's activated.
I don't even see.
No, we can hear. We can hear Dr. Lawrence.
Okay. So the biggest surprise has been the mRNA vaccines. And in fact, the mRNA vaccines are not new. The earliest papers on mRNA vaccines are more than 15 years old. And the idea of mRNA vaccine begins with an experiment where people injected naked mRNA into muscle and demonstrated mice and demonstrated that these proteins get expressed in the muscle without any further work. And subsequently, when the idea of using it as a vaccine came about, when they found that this antigen is expressed by injecting mRNA, where highly immunogenic, which is not surprising to an immunologist, but this work was done not by immunologists in the beginning. And the challenge for mRNA vaccines, one reason it didn't get funding was mRNA had several challenges. This was highly immunogenic, and therefore immunological reactions were possible. And second, the mRNAs were labile. So they are easily destroyed. Anyone who works on mRNA knows that it's very difficult to store them. So you always convert mRNA into cDNA and then you store them. So these technical challenges were overcome over a long period of time. And now we have these vaccines, which are basically chemically modified mRNAs. These chemical modifications lead to several things. One, they reduce the immunogenicity and they increase the stability. And some companies are even trying to optimize these so that they can even be stored eventually at fridge temperatures or room temperature. So it is, so basically there is a lot of technical advances which have been going on in mRNA handling, which has led to the vaccines. And so this all the other vaccines are really nothing new there, in the sense that we have been having protein, that is component based vaccines. There are even now some killed vaccines are also under development. But the key thing to know is that why did people go for these newer kind of vaccines like mRNA vaccine as opposed to traditional approaches? These are some immunological reasons for it. And even the Oxford vaccine, adenoviral vector vaccine, is also a novel vaccine in the sense that these Oxford vaccine is actually a non-replicating viral vector vaccine, which essentially is a DNA vaccine. So the nucleic acid in the Oxford chadoxon does not replicate. It delivers this genetic material into the cell. And the key difference is that when you want a DNA vaccine, it needs to be delivered to the nucleus where the mRNA can be produced and then protein can be expressed. Whereas mRNA can be directly listened, only they need to reach the cytoplasm, which is very easy, and lipid nanoparticles can deliver it there. And so delivery wise, mRNA has advantages, whereas DNA offers other challenges, which is why DNA vaccines also didn't come up that early. Rachel would remember DNA discussions on DNA vaccines in the in our seminars.
Absolutely.
Yeah. So let's come to the life cycle of the this coronavirus. You know, the coronavirus enters through ACE2 receptor. But what we need to remember that ACE2 is not the only receptor. All coronaviruses actually have a property called dual receptor utilization. That is, the spike protein has two domains which can independently target two different receptors. At any one point, one is a dominant one, the second one is an extra one. If the first one gets blocked, it tends to use the other one. So this becomes very useful at the time when coronaviruses are jumping from species to species, so that they can retain one and mutate the other. So in any given species, one is dominant, the other is free to vary, and therefore it makes it possible to pre-adapt and jump. So this, and these coronaviruses are notorious from jumping from species to species. And once it, and I've tried to highlight a few things in the life cycle which are relevant to vaccines. Once it enters the cell, and the first protein that is the expressed by the coronavirus is the ORF1, which encodes as a polyprotein which encodes the replicase complex. The replicase complex includes the helicase and the RDRP, that is RNA-dependent RNA polymerase, and this helps it make some multiple copies of the mRNA. And so this is the first protein that is expressed. Subsequently, other open ORFs and non-structural proteins get expressed, and if you look at it, number of recognized ORFs in the coronavirus, especially SARS-CoV-2, is increasing. Now it's more than 2 dozen. And the most frightening aspect of this is that most of these proteins encode immunomodulating proteins. But surprising how many of them are there. There are nearly a dozen proteins which interfere with interferons, and every trick in the book of viruses is there. So once the other proteins get expressed, the coronavirus can hide from the immune system by tricks such as downregulation of MHC class 1 and MHC class 2, which is a very well-known trick in the case of all viral infections, and this also does it. So once MHC expression is downregulated, it can hide from T cells. And once MHC expression is downregulated, the natural killer cells are supposed to be the ones which can deal with, cells which where the self tag of MHC is absent. However, studies in COVID have shown that when the NK cell function is depressed in COVID patients, So this, and so I don't remember whom, somebody called coronavirus as a airborne HIV infection, kind of describes the kind of tricks it can do that. And on the right side is that, this is the receptor usage of various types of coronavirus. And interestingly, most of the coronaviruses have uses receptors which have some kind of an enzymatic activity, such as DPP4 in the case of MERS and aminopeptidase N. So basically these are highly capable of jumping species because of their dual receptor usage, and the typically, so this is a common property. And one of the importance of this is that our spike protein, which is the most common antigen used in, they are actually expressed late. That is, first comes the replicase, then comes the non-structural proteins and the other proteins which suppress the immune system, and towards the end only the spike gets expressed. So there are some limits to using spikes as a protein in terms of immune response, the cell-mediated immune response, that you can have many cells which are all infected by coronavirus but do not express any MHC, that is antigens of spike on the surface. Then there are many ways in which our body recognizes the infections, and in the case of viral infections, there are many proteins starting from the toll-like receptors, that is TLR7 to various ring helicases, etc., which recognize the viral infections. And at every stage there is some coronavirus which inhibits every one of them. So this is the why coronavirus has a relatively large genome with large number of non-structural proteins, which are all everything from the ring helicases to even the MDA5 is inhibited by the various coronavirus non-structural proteins, and subsequently it also has downstream inhibitors of interferon expression. But one thing I want to say, which is very, very crucial in understanding how the coronavirus escapes the immune system, is TRIM21. Rheumatologists in this group would recognize that TRIM21 is nothing but Rho52. It's another name for Rho52. And what does TRIM21 do? TRIM21 is the most high affinity FC receptor in the body, but we don't think of it as an FC receptor, and nobody calls it an FC receptor. That is because this is an intracellular receptor. It's a cytoplasmic FC receptor for immunoglobulin, which also has direct viral sensing capabilities. And this is direct viral sensing capability of TRIM21 is important in the case of enteroviruses. So enteroviral mutants which escape interaction with TRIM21 are like enterovirus 71 cause invasive enteroviral diseases. So defects in this, that shows that this mutation is ultimate proof that something is a target. And then what does it do? That is, we generally think that antibodies stop infection by blocking viral entry. This is mostly true, but people have shown that even when there's always some viruses that enter even in the presence of neutralizing antibodies. So neutralizing antibodies are not 100% protective barrier. There are always escapes. They do enter the cells. Still, in the presence of antibodies to most viruses, it does not cause an infection. How does it do that? It was found that, the level of expression of TRIM21 is the rate limiting step that determines the threshold of infection for cells. This shows that what it does is TRIM21 adds an ubiquitin to the virus, the antibody-coated virus, and this ubiquitin targets to the proteasome, and it short circuits the viral infection pathway. And this particular TRIM21 mediated pathway is most important for adenoviruses and enteroviruses. And interestingly, this pathway is mostly identified when people encounter technical problems with use of adenoviruses as a the vector for gene therapy. So if you have antibodies to adenoviruses, these TRIM21 pathway will eliminate the vector very efficiently before it can do anything. So now you understand the relevance of this. This is relevant in the sense that that is papain-like protease of coronaviruses have de-ubiquitinating capability. Therefore, antibody-coated coronaviruses can be, can escape from the clutch of TRIM21. So antibody-mediated defenses are leaky. Still happens. It's a leaky defense. So you will not get 100% protection through antibodies to viruses which can escape 21. Now we remember this is one of the reasons why had predicted that there will be, you have to, no vaccine will be sterilizing. And second thing is that this also gives challenges to the adenoviral vector vaccines, because anybody who has antibodies to those adenoviruses will be very good at eliminating the vaccine vector, and therefore it may not produce antibodies. And this is a challenge that the Oxford vaccine has encountered. So two issues with TRIM21. And then interferon is not produced, which is the most important way in which our body senses the presence of the viruses. It also does a lot of other things. We know that there is a lot of cytokine activation, and this is because ORF3A, or that is one of the non-structural proteins of coronavirus, directly activates NLRP3 inflammasome. And this is actually a viroporin. And so essentially, it has been shown that even when the viral mRNA levels of infected cell is up to 50%, the interferon genes are not activated in the infected cells. On the other hand, you have IL-1 expression and then IL-6 expression. Basically, the body and cells are skewed towards an inflammatory response, and which are usually inflammasome mediated. In addition, the binding of ACE2 leads to downregulation of ACE2 and an unregulated renin-angiotensin system activation, which dysregulates the vasomotor regulation, leading to spasm and hypoxia. And hypoxia-induced factor alpha also contributes to inflammation. And in fact, in the hypoxia model, that is the high altitude sickness, there is increased IL-6 expression due to the HIF alpha pathway, and dexamethasone inhibits the HIF alpha expression. It is one of the mechanisms of action of dexamethasone, that is inhibiting HIF alpha. And HIF alpha also promotes thrombosis. This helps us understand why there's a lot of thrombosis in coronavirus infections. And we are all familiar with the antibody, that is typically antibodies coat the virus and prevents it binding to the receptor. And so antibodies are the primary modes of protection against viral infections, and those individuals with defective antibody protection, that is humoral immunity defects, lead to recurrent infection by viruses, which may even have a lifelong protection. So antibodies, if you have effective antibodies that block infection, you can get protection. Whereas the cell-mediated immune system is most important in clearing virus-infected cells. So it is a second line of defense after the antibodies. If the first line is completely effective, you don't need the second line. So, but first line will never be completely effective because of the TRIM21 escape. However, until recently, we did not have any idea how much of protection this offered. We know it is partial, how much it is relevant. And this has been answered by the Novavax vaccine, which used the protein subunit vaccine, which offers 89% protection. That means antibodies can give you 89% protection. And you need additional mechanism for the rest. And if you look at the difference between 89% and 95%, it is actually more than 50% additional protection if you look at it. So it's actually a huge difference. 89 to 90% from, if you look at it from protection, it's a huge difference, but it may not look so from our perspective. Coming back to cell-mediated immune response, we have to remember that all nucleated cells express MHC class 1 antigens, whereas only the professional antigen presenting cells express MHC class 2. And the MHC class 1 NMHC class 1 restricted antigens are recognized by CD8 cytotoxic cells. So if you have a viral infection, to clear the infected cells, you need CD8 cytotoxic response, and the hallmark of this is interferon gamma response expressing CD8 cells, which you might have noticed in phase 1, phase 2 studies of vaccines. The question is, how do you get a good CD8 response? And CD4 response is required not for killing the viral infected cells, but in providing help, that is licensing the B cells that produce antibody and licensing the CD8 cells that produce cytotoxic. Both of us require CD4 T cell response, and so this is where it comes. And one of the thing we need to remember is that CD4 cells can actually have cytotoxic activity, but it happens much later. That is, only if CD8 cells fail to clear viral infected cells do CD4 T cells come into play. And once that happens, you will get a lot of pathology. That is in fact, it is not surprising that if you look at autoimmune diseases, most of the cells doing the damage, for example, take type 1 diabetes, it is CD4 T cells do the damage. So CD4 T cells are not, it is like sending the, literally like bombing the place. And in the case of coronaviruses, there is not much of a correlation. If you look at extrastructurally between the presence of the virus and the CD4 T cell infiltration, and so the tissue damage is not surprising. And another consideration in the case of coronavirus vaccine development was reports of antibody-dependent enhancement in some of the animal models. So what is antibody-dependent enhancements? That is, if there is an antibody which bites the virus but is not neutralizing, these antibodies may actually help the virus enter the cells, especially immune cells like monocyte macrophages, which express Fc gamma receptors, and the virus can enter the cell through Fc gamma receptors and may infect cells which may normally not be infected by the virus and cause enhanced pathogenicity. And this is well described in the dengue. And there were reports of ADE in the case of SARS and MERS. So this is why all the efforts focused on narrowing the focus to the receptor binding domain and neutralizing antibody. And second mechanism of enhancement is that formation of large number of immune complex may directly induce secretion of cytokines. And another thing unique to coronavirus was that when whole virus vaccines were tested in animals, it led to increased lung pathology. This lung pathology consisted of eosinophilic infiltrates, and eosinophilic infiltrates suggest a Th2 biased immune response and Th2 biased immune response. So that is why all the efforts were at developing an immune response which was Th1 skewed, that is away from Th2 response. So this was a major consideration. So what do you want? You want antibody which is neutralizing. We want a CD8 response. Hello? We need a CD8 response, and we want a Th1 skewed immune response. So let's look at how do we get, as well as getting antibody response, it is not that very difficult because, if you use an adjuvant and a protein, you will get antibodies. So the challenge was, in the case of coronavirus vaccine, was getting a good CD8 response. So let's look at how the antigens are presented. Basically, there are two main pathways for antigen presentation. You have the endogenous pathway and the exogenous pathway. Endogenous pathway is where the proteins which are expressed in the cytosol, that is essentially when proteins are degraded in the proteasome, these peptide fragments from the proteasome are loaded to MHC class 1 molecules and expressed on the cell surface. So essentially, if you the MHC class 1 like presentation or presentation of antigens that induce CD8 response follows a cytosolic pathway, and this has a preference for proteins which are expressed in the cytosol, that is endogenously produced antigens. So how do you get and on the other hand, the CD4 response comes from that is exogenous pathway, that is proteins which are endocytosed by the and the lysosomal pathway, the proteolytic degradation in the lysosomes leading to loading of the MHC class 2. So there is of course exception to this code is called cross presentation, but so what do we need to do to get a good CD8 response in an efficient and predictable manner? You need to express the antigen inside a cell. How do you do that? The best way to do it is, the traditional way to do it is using attenuated viruses, or attenuated viruses. Virus expresses proteins, and these proteins, some of it will go into the proteasome and they will get. So the surest way earlier was live attenuated vaccine. But live attenuated vaccine take many years because they have to prove it safe, and therefore live attenuated vaccines are a poor candidate in a pandemic setting. In traditional way, when you could wait 5 to 10 years, that is okay. So you needed platforms which are proven, which is where vector vaccines and then the nucleic acid based vaccines came into play. If you are basically, what do you need to express a protein? You need to deliver the information, the message in the form of DNA or RNA. DNA or RNA will naturally express the protein inside the cell, and they will get presented through the endogenous pathway. On the other hand, if you are just delivering a traditional protein, that is, you are delivering protein, these proteins will be presented by the exogenous pathway, and they are not very good at producing a CD8 response. They will produce CD4, and now the question is only to skew it to Th1 response. But CD8 response are poor in the case of subunit vaccine, skilled vaccine, etc. So this is why these choices are there. Now coming to adjuvants. Adjuvants were the big secrets of immunologists. For until the late 1990s, nobody talked much about adjuvants. What is ILM doing? What is to get different kinds of immune response? You added various things. What do they do? The most importantly, when a protein is cross-linked and then there's a multi-antigenic cross-linking, it leads to greater immunogenicity. So many of the older adjuvants like mineral oil, they just precipitated and formed complexes which induce, but besides that, they also activate various innate. Now we know more about innate immune system. And they activate innate immune system, they produce cytokines, they increase co-stimulation, and some of the best adjuvants that can induce Th1 response are the TLR agonists, such as, for example, imiquimod is a classic TLR7 agonist, and produces potent interferon response and Th1 response, and DNA, that is prokaryotic DNA from which the CpG is derived, they are potent TLR9 agonists, which also induce a strong Th1 response. So why this is relevant is that these mRNA and DNA vaccines, they are potent stimulators of these TLR7 and 9 and therefore are naturally potent adjuvants in themselves. That is, you don't need additional adjuvants if you are using an mRNA or DNA. In fact, the challenge is to reduce their immunogenicity so that they don't stimulate it too much. And on the other hand, the other traditional adjuvants have a problem that they produce potent antibodies, but they don't help you produce a Th1 immune response. And this is why the Indian killed vaccine, Covaxin, they use a TLR7/8 agonist, which is related to the imiquimod. It's not exactly imiquimod. Some code is there, chemical formula, we don't know. But what is to be remembered is that these are, even the Covaxin is a novel vaccine. And lot of vaccine related advertisement may be related to the use of adjuvants. So when we are doing a vaccine trial, we are interested in the safety of the adjuvants. What can adjuvants do? Adjuvants can stimulate the immune system. And we know that there are, an immunologist would know things like adjuvant-induced arthritis. You inject an adjuvant, potent adjuvant, you get an autoimmune arthritis. And if you inject imiquimod, in fact, imiquimod is used in skin disease like warts, and some of the adverse events are autoimmunity. You can get subacute cutaneous lupus-like lesions if you give imiquimod. So these potent adjuvants have the potential to create autoimmunity. So this is a concern. So this is why there is everyone was concerned about reports of very rare, however, of demyelination, etc. in the case of certain vaccines. But while we have to remember these are extremely rare, and this should be a consideration. Would you give these vaccine to persons with autoimmunity?
Dr. Lawrence, sorry to interrupt.
Yes.
We have a lot of questions that we need to go through.
Yeah,
so we would. It'll be great that we can, save some of the slides for you. Thank you.
Yes. I basically want to provide the background to any questions that would come. That's the purpose of the thing. So therefore, summarize, if you look at it, these vaccines, the choice of the mRNA DNA vaccines is obvious now. That is, they provide potent naturally Th1 skewed immunity. They're good at inducing a CD8 response, and all of them would produce CD4 response, should reduce CD4 response. Whereas the protein-based vaccines are very good at producing antibodies, but you can't skew them to Th1 or unless you use agonists like the TLR agonists, which have potential safety concern. And finally, so that is a summary of this. And these phase 3 trials and post-marketing surveillance are most important for safety, understanding, because vaccines produce side effects which are rare. However, when you are giving to healthy people, even the rare side effect which happens one in 10,000 or 1 in 50,000 is actually relevant. So that's why even after the phase 3 trials are over, it is important to continue monitoring subjects. And then after that, maybe case control study, we need to look at background rates, etc., understand if a particular adverse event is actually related or random. Then one of the common questions which everybody tends to ask me is, if you have a COVID infection, do you need is vaccine immunity better than natural infection? The answer is definitely yes, for several reasons. The most important thing is that when you have COVID infection, for various reasons, there is a profound defect in the germinal center formation. There is no germinal center in the lymph nodes of COVID-infected subjects, and this is due to inhibition of follicular helper T cells, which are necessary for the germinal center formation, and the antibodies formed are not from the germinal centers. And it is in germinal centers that somatic hypermutation and affinity maturation happens. So if you, and also only through germinal centers and affinity, you can get long-lived plasma cells. So long-lived plasma cells are required for long-term production of antibodies. So everyone is familiar that after the COVID infection, the antibodies wane fast. That is because there is no germinal center formation and the antibodies are not that particularly great. Whereas in a vaccine, there is normal follicular helper T cells and there is normal germinal center formation, and affinity maturation can take place, and you have better antibodies which can potentially last long. But we can't answer this yet because nobody has, I don't think anyone who has got the vaccine has had more than 6 months duration. So time will tell how they will last, but anyway, it will be better than natural infection. And again, one of the questions somebody has asked is, where are we going from here? How is it going to end? So I said this somewhere in January itself. Eventually this, basically, how did I say that? Because I looked at in January when it was clear that this coronavirus emerging, everyone was focusing on SARS, but I was looking at the entire gamut of coronaviruses in animals and humans, etc. And from, all kinds of, so the thing is, there are several, if you look at the human coronaviruses, human in inverted commas, You have the OC43, HKU1, and the NL63 and the 229E. And most of them cause childhood infections. And everybody says they're common cold coronavirus. But that's a misnomer. In fact, if you look at series of people with serious intensive care units with viral infections, a lot of them have, it can cause sporadic severe LRTI even in elderly, and only the HKU1 causes only the upper airway infection because its receptor is present only in the upper airways, and they are not actually that benign. So when it occurs sporadically, nobody notices it. But during outbreaks in non-immune populations, high levels of mortality have been observed. During an outbreak of OC43, 8 out of 98 died. It is higher than the SARS-CoV-2 and is on par with the original SARS. So in non-immune subjects, none of this is actually benign. So why don't we have serious infection? Because most of these coronaviruses infect people in childhood, and then your subsequent infections are actually, reinfections in immune individuals. So essentially what it shows is these coronavirus do not naturally, the human coronavirus do not produce lifelong immunity. And the first immunity in the children are mild, just like we are seeing in SARS-CoV-2. And there is a considerable variability in severity, with majority being mild or asymptomatic and some people getting serious, and very much like the SARS-CoV-2. So SARS-CoV-2 is not really an outlier. Our real outlier was SARS. For the reason specific to SARS, it caused it didn't cause any asymptomatic infections, and therefore it did not become a pandemic. And so we could expect that these would also eventually become like an endemic human coronavirus. And the best example is OC43. Genomic comparison of OC43 showed that this is closely related to bovine coronavirus. And bovine coronavirus is the commonest cause of pneumonia, that is infective pneumonia in cattle, and it's very common when cattle are being transported in mass. And the molecular clocks have shown that this transmission most likely, that is from jumping from cows to human, took place somewhere around 1890. And interestingly, there was a flu pandemic of 1889, 1890. But at that time, nobody knew about viruses. We don't have any samples to confirm the identity of the causative agent causing 1890 pandemic. It might have been this, or it might have been something else. So,
okay, thank you very much, Able. I know a lot of people just talk about coronavirus, but you really need to have an in-depth knowledge of immunology to really understand and explain it in such great detail. And thank you very much. I have a